Hello everyone, my name is Ayush Patil. I would like to express my thoughts on the following topic, which is touch DNA, revolutionizing evidentiary DNA forensics. Touch DNA particularly refers to the availability or the presence of DNA which is transferred through direct cells. Touch DNA is also known as trace DNA. It is a concept in forensic science which helps and enables the investigator to collect the minute traces of DNA which has been left by the person at the crime site or crime scene. According to the basis of foundational Locard's principle, a person when he or she comes in contact, the skin cells are left, which under that there are epithelial cells which consist of DNA. By analyzing the DNA, the forensic scientist can help to establish the legal evidentiary linkage between the suspect and crime scene, and it also adds additional information into it and excludes the innocent individuals. Let's move on to the modes of lifting touch DNA samples. First is the swabbing technique. In this, cotton swabs are used. Second is tape lifting. Many jurisdictions routinely employ the tape lifting method. In this, seen, safe, fast TM mini tapes have been used to increase the collection of DNA. Third is the extraction scrapping method, and the fourth is the FDA card. There are types of touch DNA extraction, which includes organic extraction. Second is the automated DNA extraction. Here the question might arise: how the DNA has been isolated from the cells. So here isolation comes into the picture. DNA is extracted from the cells, where in touch DNA small amount of samples are used, and here it has been focused for maximum extraction. So the general steps includes the first, cell lysis. The cells undergoes or it's subjected to the lysis buffer, where the cell membrane have been broken down and the DNA has been released. Second is the purification of DNA. After the cell lysis, basically here the DNA is been separated from cellular debris and it further undergoes DNA purification. And after the isolation of DNA, it undergoes amplification. Amplification is the process of producing millions of copies of DNA. In this, there are some steps. First, denaturation. Second, annealing. Third is the extension. So this three undergoes a cycle for 20, and this cycle has been run for 25 to 30 times, which leads to formation of a DNA sequence. And it further undergoes, after this amplification, it undergoes capillary electrophoresis. In capillary electrophoresis, the DNA fragments based on the size have been separated, like there's a gradient which has been created, and we also use fluorescent dyes over here. Let's move on to the concept, which is next generating sequence. Next generating sequence consists of there's a vast sequence of DNA which creates a high DNA profile. It has some kind of applications, some examples like criminal casework, DNA phenotyping, or etc. So let's move on to the benefits. First is the specificity. Second, the being a fast and efficient method. Third, versatility, applicable to different surfaces. Fourth, like with the advancements and research, the sophisticated kits and equipments are well developed. With these benefits, there are some kind of limitations. First is the variable deposition. In this, the amount of DNA can be which has been collected can be vary widely based on the surface and texture. Second is the DNA degradation, which can occur due to the exposure to the temperature or UV radiations. Third is the DNA destruction. Contamination. Contamination can occur due to the use of fingerprint brushes or due to the handling errors. And the fifth, trained personnel, or the last, the false interpretations. The major challenges are regarding to the sampling and handling, and basically contamination is also a challenge. But this contamination can be mitigated with the use of proper equipments, care, and following proper protocols. With this, I would like to conclude saying this, that it offers the new avenues in solving crimes. It has been revolutionizing to the forensic science. According to various publications or the articles, it tells that there's a loss of DNA samples for around 90%, while some says it's around 39%. So with this, it plays a vital role in criminal justice, and which is undeniable. Thank you.